So for those of you who might not be familiar with the organization, Past Global Changes, or PAGES, we are an international group that coordinates and promotes past global change research. Much of our science is done within working groups that cover a variety of topics and timescales. I invite you to visit our website for more information, and Sarah will put the website in the chat for everyone. And you can read more about everyone's research in our working groups and how you can get involved. So this is the first of a series of webinars about PAGES endorsed and affiliated groups. If you are interested in receiving more information about PAGES webinars, please go to our website and join our people database, and you can sign up for e-news and keep an eye on all of the events that are happening with the events calendar. So with that, I'm going to introduce Ariane. Her research activities are varied, but focus on human-environment interactions during prehistory. She's interested in interdisciplinary approaches and modeling techniques as a means of exploring the impact of climate change on human systems and the influence of landscape structure on human population dynamics, gene flow, and patterns of cultural evolution. So she is interested in spatial analyses, archaeological survey, spatial cognition, and wayfinding. So as you can see from her title, today she's going to be telling us about deep time human environment interactions. And with that, I will turn it over to her.
Thank you very much, Jacob, and thank you for the invitation to speak to PAGES members. The research I'm going to talk to you about today is part of a research program that the Hominin Dispersals research group has been carrying out for the last 10 years or so. And we're a group of archaeologists, paleoclimatologists, and paleogeographers. And as you said, we're really interested in studying human-environment interactions and understanding, among other things, human decision-making on a landscape during times of climate change. A quick slide showing the complexity of the research program, which I'm only going to be showing you work package 1.1, the least glacial maximum, because that's the one that we've published to date. So as part of our interest in working from a multidisciplinary framework, we're very interested in integrating anthropological and archaeological hypotheses into the model building process. And one of the things that we're interested in this particular research in investigating is what the impact of the distribution of suitable habitat might have been on the distribution of human populations in the past, specifically during the late glacial maximum in Europe, and what knock-on effects that would have had on their genetic structure and ultimately on the patterns of cultural transmission. So we know that humans are a highly dispersible species. That sort of is an obvious observation, but that doesn't mean that they're evenly distributed across the landscape. So our first goal, therefore, is to try to determine what the distribution of suitable habitat would have been during the last glacial maximum, which begs the question, what constitutes suitable habitat for human beings? We're talking about a time frame when people were foragers or hunter-gatherers, if you prefer. And we know today that hunter-gatherers are perfectly capable of adapting to a very wide range of environmental conditions, from the Arctic Circle to the Kalahari Desert. And they do it using a combination of social strategies, mobility strategies, technological innovations, and also physiological adaptations. So it would be disingenuous to claim that temperature envelopes, for example, are the only constraints on the distribution of human habitat. A more balanced approach, we feel, is required to define habitat suitability, and one that considers both social and biological factors. So if we look at anthropological data, I'm thinking of ethnographic data and specifically, we see that hunter-gatherers actively evaluate strategies that are designed to minimize risk over the long term. They're very sensitive to ecological risk. The ability to deploy a variety of strategies to mitigate ecological risk is deeply embedded in their social, technological, and economic factor. They anticipate and actively evaluate strategies that minimize risk over the long term. And we want to integrate that knowledge into our model building exercise. We have a second hypothesis that grows out of this information that we want to test, which is that climate variability, rather than simply temperature minima and maxima and precipitation minima and maxima, for example, is a potential risk factor. It introduces uncertainty or stochasticity into the system that should be integrated into the research design. And it's something that we wanted to test. So our research design is to model habitat suitability doing the LGM. We're going to use the archaeological record. We produced high resolution climate models to produce the climate variables, which allowed us in turn to derive climate variability measures, which were included as candidate predictors. And then we used other climate proxies to test climate model outputs. All of this was then used to test the impact of habitat suitability using agent-based models on population structure, on gene flow, and on cultural transmission. This is the distribution of archaeological sites that were used in our modeling. We had just under 100 sites that were securely dated using radiocarbon dating to the last glacial maximum. You'll notice that the distribution of these sites isn't even. It's uneven across the landscape. And so a particularly interesting thing to note is that one of the supposed glacial refugia, the Iberian Peninsula, which together with the Italian Peninsula and Balkan Peninsula were supposed to be southern refugia for temperate species, which humans are considered to be. And yet the center of the Iberian Peninsula is demonstrably empty of archaeological sites that are securely dated to this time frame. So all of this is something that we're going to investigate in our research. The statistical, so the climate model that was produced using a global model, a GCM, was subjected to a GAM, general additive modeling, to downscale it so that we had climate data that was produced at a scale consistent with humans' daily foraging ranges, that is about 10 to 15 kilometers. And also at monthly time steps, which allows us to look at variability at subannual scales, which corresponds to the frequency of residential moves for most hunting and gathering people. And this is the publication in which Guillaume Latombe and colleagues from the Laboratoire des Sciences du Climat et de l'Environnement described the downscaling exercise and the types of interpolations that we use to produce the surfaces from which we could extract climate variables for our modeling. Recently, members of the group produced an article that compared climate proxies, and this was biogenic isotopic signatures from animals provided from archaeological sites, and compared those to the downscaled climate model data. And we found pretty good agreement between the climate proxies and the model outputs, which is reassuring. We'd already done the modeling. So back to the archaeological model, we used 52 candidate predictors, which included fairly traditional climate variables, precipitation and temperature, the minima, the maxima, the averages, monthly, seasonally, and annually. The climate variability indices, which included standard precipitation index and standard temperature index. Okay, so the technique we used for the modeling was random forest. So random forest is particularly useful for handling the types of complex non-parametric data that we have. It's a non-parametric tool for variable selection and regression. It has the ability to deal with large numbers of predictors that have relatively few observations, and you'll remember the slide with the number of archaeological sites based on the domain, spatial domain we're looking at. We really didn't have that many observations. It can handle nonlinear relationships, and a lot of the relationships with the climate variables are nonlinear, and it produces useful measures of global performance, which we'll be using in the next slide. I'll explain how we use them iteratively. There we go. So this is a table that shows successive steps, successive modeling steps. And for each iteration, we measured variable importance and model performance. And at the end of each run, we removed from consideration the least important variables, so 10% of the least important variables. We started with 52 variables, and we ended up with I think in our best model, there were 6 variables. So every time we remove the less important variables, we rerun a random forest and we look at model performance. And when model performance starts to decline relative to the number of variables that we've selected, we determine that that's the best model for us to retain. And in fact, the best model that was retained is model 26 here, down here. Oh, down here, showing you on the wrong screen. There we go. This was published in 2017, an article called Risky Business, the Impact of Climate and Climate Variability on Human Population Dynamics. The results demonstrated that climate variability is an element of ecological risk that was being considered by people when they selected areas on the landscape to live in, because several of our measures of climate variability were selected for during the iterative random forest modeling exercise. Now, there's a potentially confounding factor, which is that humans use different mobility strategies as a means of minimizing risk. So they don't just react to the landscape, they also mitigate what the landscape might be constraining in terms of their choices. And they do it by using different mobility strategies. And there's a spectrum of possibilities. And at either end of the spectrum, you have what's called residential mobility, and at the other end, logistical mobility. So the residential strategy means that people basically extract resources from their immediate environment from one residential site. And when that catchment is depleted, then they will move to a new catchment, a new site. So residential relocation. And the logistical strategy allows them to expand the catchment around an archaeological site by sending work parties out. And the work parties might go 1 or 2 days walk away from the residential site, gather resources and bring them back. And it's a way of extending the daily exploitation catchment around a site, and reducing the number of residential moves that are required, which are, of course, energetically expensive things to do. And environmental conditions should logically affect the choice of mobility strategy. So as resources become depleted, you should shift to a logistical, logically, you should shift to a logistical pattern of mobility if your goal is to not have to relocate as often. So we decided to test that, knowing that one of our members, Julien Riel-Salvatore and Michael Barton in 2004, had shown that the frequency of lithic retouch in an archaeological site, so that's the frequency with which stone tools are resharpened, leaving little chips in the assemblages, is in fact linked to mobility patterns. So it can be used to predict which mobility strategy was being used by a group. So the less logistically organized foragers are not going to have logistic campsites, and most of the sites that they occupy are going to have fairly high retouch frequencies. And the logistically organized hunters and gatherers will have low retouch frequencies because the logistic campsites that they produce are occupied for short periods of time, and they don't do a lot of retouching of their tools. So we conducted a test. We're looking at the relationship between habitat suitability predicted by our model and retouch frequency. And one of the interesting things we noted is that if you look at the higher suitability regions, so all of the sites, all of the occupations, there were 43 of them, where people were living in relatively high suitability regions, had low retouch frequencies. So they were actually using a logistic strategy and fairly tightly constraining their mobility to that type of strategy. But if we look at the people who were living in parts of the landscape that were both high HS values, but also low HS values, we see that they're, that had overall low habitat suitability. We see that they had high and low retouch frequencies, indicating that their mobility strategies were quite varied. So one of the interpretations of this could be that as people move into less desirable areas where there's a higher probability of being wrong in your predictions about resource distributions, that you might be testing out a wider variety of mobility strategies, possibly on a seasonal basis. And that's the article that was. So the more stable, logistically organized hunter-gatherer groups were correlated with high habitat suitability values, and the more varied mobility strategies seem to have been adopted by hunter-gatherers in low habitat suitability regions. So the next step in the analysis was to use the habitat suitability model to fuel an agent-based model that could explore how the distribution of suitable habitat affects population dynamics through an impact on genetic structure and cultural transmission. So the initial condition of the habitat suitability map is our habitat suitability model. Sorry, the initial condition of the agent-based model is the habitat suitability model. One of the first things we did was initiate the model with roughly 6,000 agents that were distributed in the landscape, and we gave them 250 time steps. Each time step corresponds to about a month to distribute themselves across the landscape, and basically let the agents determine where they were going to cluster. We gave them a weighted random walk algorithm for determining where to go that favored the high HS suitability cells. And the result was clustering into 6 major regions, which are shown in color here, that correspond roughly to a major part of France, part of Cantabria here, which we call Spain in this set of runs, Portugal, Valencia, so you can, Italy, and a little bit of the Jura, which includes Central Europe and Switzerland and northeastern France. So 6,000 agents randomly distributed on the landscape and allowed to run for about 250 years worth of time steps before being tagged with a genome. And that's going to be important because we want to observe gene flow between agents after once the experiment gets running. So that's how we set up the initial parameters for the test. The agent-based model itself is composed of agents, and the agents are actually family groups rather than individuals. Partly because that's expedient, partly because their family groups would be the sort of natural minimal social unit that people would have been moving within. We then have algorithms to control their mobility. So in the next slide, we'll describe how we constrain their home range and also the radius of movement for agents from their home cell. And we programmed in a population growth rate, which is the result of the interaction of the probability of agents producing new agents and the probability of agents disbanding or dying. And at each sort of set number of time steps for each of our experiments, we looked at the final population size per region, and we looked at the genetic makeup of the agents that lived within the region. So a little word on mobility. Our agents must move at every time step. The cells are weighted as a function of their HS value relative to the distance from the agent's current cell and within a predefined radius. And we ran 3 different experiments, a radius of about 50 kilometers from the home cell, within which agents can make a decision about where to move in the next time step, a radius of 100 kilometers and a radius of 200 kilometers. And that corresponds to, well, the 200 kilometers corresponds to an average distance that can be, that is documented in their archaeological record. If we look at lithic, the movement of lithic materials across space for the Upper Paleolithic. Now, the life cycle for our population is determined by 2 variables. One is the probability that agents produce other agents, probability X, and that is kept constant. The offspring of a reproduction event will have a blended genome, half of the genes from one agent, half of the genes from the other agent. And remember that those genes have been sort of baked into the model in that very first initial step of the model. Important for archaeologists to note that we didn't introduce any kind of cultural mediation on reproduction. So any 2 agents have the same probability X of reproducing if they meet after a residential move during one time step. Then we have agents that have a probability of disbanding or dying, and that's a Y or Y variable. And that is something that we also experimented with. So we're experimenting with radius of movement, and we're experimenting with the probability of disbanding. And what we did with the probability of disbanding is test different effects of habitat suitability on that parameter. And since X and Y affect the net growth rate of the population by varying Y and keeping X constant, we're allowing habitat suitability to affect the net growth rate of the population. So you see here is the straight line where habitat suitability is 0. In other words, it doesn't matter whether a cell is low or high habitat suitability, the probability Y remains constant. And then we start affecting in each of the experiments how favorable or unfavorable HS can be in terms of its impact on the probability of disbanding. So that's how we're using HS to affect growth rates. The results were published in 2018 in an article in PLOS One. And here's the first slide that shows some of the results. So in blue, from the lightest to the darkest, we have HS that has absolutely no impact on the probability of disbanding and thus on net population growth, and HS that has a multiplier effect on the probability of multiplier, so it has a lower effect on the probability of disbanding and a multiplier effect on the probability of survival. And you see here that net population size for the high benefit groups, so when HS is really influencing net population growth, the population rate grows quite quickly and then stabilizes in all 3 of these experiments where the mobility radius was being changed. So all things being equal, in other words, when HS has no effect, the population never reaches maximum carrying capacity predicted by the number of high habitat suitability cells in a population's territory. So those are that flat line in pale colors. That's all things being equal. HS doesn't have an effect on population growth. As HS has greater benefits, the more it affects our Y parameter, the population increases, irrespective of the foraging radius. The second observation from this graph is that as the foraging radius increases, the population also increases for HS benefit anywhere above 0.5. So we've got a threshold value here beyond which the population is going to reach carrying capacity. In this graph, we're looking at regional population sizes, and you can see that they're linked to the size of the core region. If you remember the very first slide, we looked at how we divided up Western Europe into different core regions based on the cluster of high suitability cells. And the largest of those core regions was the sort of central French and northeastern Spain region, and that's the one that shows up in purple here. So you can see that regional population sizes are linked to the size of the core region, and that the largest core region is always going to be favored, irrespective of the different mobility radiuses we test, 50, 100, 200, and different HS benefits. So we're playing with 2 different parameters with these different experiments, and we always see that this central region, which we're calling France in this graph, is favored. What happens is the smaller regions will be more or less favored depending on the radius of mobility. This has an effect on the genetic makeup of the population. So in this graph, we're just looking at the proportion of genes from different regions within a regional genome. So for example, the Portuguese genome here is composed of genes that have been inherited from individuals that had migrated in from France, from Italy, from the Jura, etc. And what we see in this graph globally is that once we have higher, if we allow our agents a higher radius of mobility, there's a lot more mixing going on between the regions, and no one region, all of the regions have got quite a strong input from the Central French region. As the foraging radii increase, regional populations are more similar, and increased gene flow occurs between the regions. So that makes sense. Italy, which is shown in the right of all of these graphs and is red, so the Italian genes show up as red, at a low radius has got a relatively stronger regional genetic signal, and that's because it's less well connected geographically to the other core regions. Now, if habitat suitability didn't have an impact on population structure, as we see in the left graph, where red is the frequency of occupation by an agent over time on the territory, and black is the absence of an agent, you can see that in this left graph, if habitat suitability didn't have an impact on population structure, the populations would be more evenly distributed, but we know from our previous graphs, the net population size would be well below the estimates derived from the archaeological data. On the right, we have the opposite end of the spectrum, maximal benefit, maximum survivorship conferred by high habitat suitability, and you see very high clustering in the more favored larger central core areas. So we conclude from this that habitat suitability had an impact on population structure, and that it was a positive one, and that it did affect the distribution of populations, since it's only as habitat benefit reaches that threshold value I was just talking about that we start seeing population sizes that are consistent with the estimates derived from archaeological data and also from ethnographic modeling based on the size of the region. So we'll conclude from this that our central region is the most intensively occupied, as opposed to previous hypotheses that suggested that it would have been the southern peninsula that were more intensively occupied during the LGM. And that this occupation reflects the location of a large and persistent glacial refugium, and we suggest a source population from which smaller regional populations could be replenished should they become extinct, which is something that we have in fact seen in several of the successive experiments that were run. I don't know why I have it. Oh, here we go. So some of our conclusions: mobility doesn't have a big impact on population size, but low mobility produces stronger regional signals, which seems very intuitive. Low growth rates produce unviable population sizes in smaller regions. The overall pattern is one of interregional genetic exchange. And Italy appears relatively poorly connected to other Western European regions. This leads to some predictions. We predict, for example, that interregional gene flow between all of the core regions should exist. That the dominance of genes from the central region is going to, which acts as a source population, should also be visible if we get paleogenomic data. That it's possible that genetic diversity was lost. For example, some of the regions like Valencia, which is on the east coast of the Iberian Peninsula, which is a relatively small core region, in several of our experiments, the population there would become totally extinct. So there is a possibility of loss of genetic diversity from these smaller, less well-connected regions. And if mobility was low, Italy should retain a relatively distinct regional genetic signature. This is all consistent with paleontological evidence for the emergence of a modern European morphotype after the LGM, and some early paleogenomic results that have been published for large-scale population reconstructions across Europe during the LGM, although that's still in its infancy, and we have yet to collect enough data to be able to see whether our predictions are correct. This is going to have some impact on cultural transmission. In archaeology, there's a hypothesis known as a cumulative culture hypothesis, which basically states that demographic processes are going to affect rates of innovation and transmission, cultural innovation and cultural transmission. Population size and connectivity between populations are the key parameters of this process. And since our results suggest a large and well-connected population in the central region, this is the population that should favor cultural transmission and innovation, both in that region and across Western Europe, because it's connected with all of the other subregions. And this is the research that we're currently undertaking as part of this work package. So I think I may have spoken quite quickly. I'm already done, and I would very much like to thank you all for attending the talk, and thank Pages for sponsoring this talk. Just like to say that our research is supported by the Fédération Québécoise de Recherche de Sciences et Culture, that climate models were produced by the LSCE, and that various people such as Matt Harris and Ben Marwick were instrumental in helping us devise the agent-based model. So thank you very much.